Welcome to Vivid Botany. We are discussing a question here about the restriction endonucleases. You said it here, restriction enzymes. That is, actually they are restriction endonucleases. So, restriction means these enzymes are produced by bacteria for the defense against the viruses. So they try to restrict the growth of bacteriophages. They try to restrict growth of virus, that is viral DNA in bacteria. That is the major function of the restriction. And next, endonuclease. Endonuclease means these enzymes cut DNA at specific site. The specific site is nothing but the palindrome. The palindrome is that which is read same in opposite direction. And nucleases are those which degrade or which digest nucleic acids, which degrade or which digest the nucleic acids. So the first isolated REN, that is restriction endonuclease, is HIND2, HIND2, Roman 2. It is Haemophilus influenza by a scientist called as Hamilton O. Smith. It was a scientist. So what they do generally, restriction endonucleases, I have explained. Restriction means they restrict the growth of virus. Actually, when a bacteria was attacked by virus, the virus will inject its DNA inside the bacteria. Then the bacteria will synthesize these enzymes, restriction endonuclease, against their viral DNA to digest the viral DNA. So they are named as restriction endonucleases. Nucleases are two types. Nuclease means a group of enzymes which will digest the nucleic acids like DNA and RNA. If they digest the DNA, they are called deoxyribonuclease. If they digest the RNA, they are called ribonuclease. So these nucleases are two types like exonuclease and endonuclease. Exonucleases are those which cut the DNA at the ends. Endonucleases are those which cut the DNA at specific sites. The specific sites are called palindromes. The palindromes are those which are same when you read them in opposite direction. I give the example for palindrome of EcoR1. EcoR1 is a restriction endonuclease. It E for Escherichia, CO for coli, R for substrate, and 1 for the order in which it was isolated. So it was isolated from Escherichia coli bacteria, we know that we commonly call it as E. coli. So E. coli R1, it's not I, it is ECO R1. 1 indicates the Roman numerical, that is the order by which it is isolated. So the ECO R1 will cut the palindrome in DNA GAA TTC. So other strand of DNA will contain GAA TTC. This is 5 to 3 and this is 3 to 5. So this is called as palindrome because when you read it in opposite direction from here to here or from here, it will be same like GAA. So this ECO R1 will cut in between G and A from 5 to 3. It will cut in between G and A from 5 to 3. As a result, here two cohesive sites are formed. Two cohesive sites are formed here. So these cohesive sites will be like this actually. This is one DNA strand and this is the other DNA strand. So these two are readily united when they are brought together. So that they are called as cohesive sites, or they are called sticky ends, or they are called cause sites also. So what is the function of the DNA ligase, right? So one more thing here, the restriction enzyme will cut in between G and A. What does it cut exactly? It will cut the ester bond in between sugar and phosphate. Here in between G and A, sugar and phosphate ester bond is present. That bond is cut here. Here also, when ester bond is cut, so two ester bonds are cut on two strands of the DNA molecule, of the double-stranded DNA molecule. So here one ester bond, here an ester bond. Both the ester bonds are cut by REN. REN in the sense restriction endonuclease, right? So what is the function of DNA ligase? DNA ligase will join the ester bond. It catalyzes the formation of the ester bond. So here, where it is cut exactly, at that place, the ester bond is joined in between the sugar molecules of adjoining nucleotides. So this ester bond is catalyzed by DNA ligase. This ester bond is also catalyzed by DNA ligase. So as two ester bonds are cut by restriction endonuclease, two ester bonds are ligated by, joined by DNA ligase. So we can say that restriction endonuclease will cut the ester bonds, DNA ligases will join the ester bonds. So restriction endonuclease are called as molecular scissors, whereas DNA ligase are called as molecular glue. So after joining the two DNA strands, it appears like this. Like this. So here the bond is joined, here the bond is joined. Both the ester bonds are joined here by DNA ligase. Then let me say the common features in between the DNA ligase as well as the restriction endonuclease. I write here REN, and DNA ligase is called as molecular glue. It is called molecular glue, whereas restriction endonuclease is called as molecular scissor. It is called as molecular scissor. And another difference is that DNA ligase will catalyze formation of ester bonds. Formation of ester bonds, whereas restriction endonuclease will catalyze breakdown of ester bonds. So one will break down the ester bond, one will reform the ester bond. Then what is the another difference in between DNA ligase and restriction endonuclease? Restriction endonuclease are very specific where they cut, that they won't cut all the DNA strand fragments from one end to the other. They very specifically they cut. Very specifically they cut. But DNA ligase are not like that. So they can readily unite the two ester bonds. They are not specific. So these are analytical points about the DNA ligase and restriction endonuclease. Now let's have a look at the DNA replication. In DNA replication, we know that one DNA gives rise to two DNA molecules. It's also called DNA synthesis, DNA duplication, semi-conservative model. It has got so many names. It is done in synthetic phase of interphase. And here, what happens? Let us suppose this is DNA strand, double-stranded DNA molecule. Remember always, the DNA does not unzip from one end to the other end like we unzip a bag, because unzipping of two DNA strands of DNA strands of one DNA molecule is very difficult as it requires a lot of energy. So that what happens here at particular point that is called as origin of replication. At that point, the two DNA strands are separated. The separation of two DNA strands is done by an enzyme called helicases, and the separation of two DNA strands is called as replication fork. It is called replication fork. Let us suppose this is the direction of replication fork. It is the direction of replication fork. So, in this direction, the fork is opened. Let us suppose this is fifth end, this is third end, and this is fifth end, and this is the third end. We know that when two DNA strands are separated by helicases, the two DNA old strands act as templates. So, this acts as template because it is the old strand, and this also acts as template as it is also the old strand. So for every template strand, a new strand is formed by DNA polymerase, and DNA polymerase will polymerize the new nucleotides only in 5 to 3 direction. That is the most important point where it creates a complexity. DNA polymerase is an enzyme. In fact, it is polymerase 1. It polymerizes nucleoside triphosphates. I am writing nucleotides in 5 to 3 direction only. It doesn't replicates in 3 to 5 direction. Remember always, replication of DNA, transcription, translation, all that takes place only in 5 to 3 direction. They never takes place from 3 to 5 direction. So let us see how the new strands are formed. This is the old strand, and fork is also opening in the same direction. So the new strand will start forming here, and it will move upwards the way the fork is opened. So this is called as the new strand. So this strand is continuously formed in 5 to 3 direction, that is complementary to the old strand, template strand. So this strand is called as the leading strand. It is called as leading strand, or it is also called as continuous strand. It's also called as continuous strand. It is called leading strand. It is called continuous strand. And for other template also on other side, another DNA new strand should form. But the problem is that towards opening of the fork, the new strand that is formed is continuously formed, whereas the other strand is not continuously formed because it's not opened readily. So what happens here? The first strand is formed here in 5 to 3 direction. This is second strand is formed here 5 to 3 direction. The third strand is formed here 5 to 3 direction. So you can assume here this is fifth end, third end, fifth end, third end, fifth end, and third end. So these strands are formed discontinuously. They are not formed continuously. They are called as, and these strands are called Okazaki fragments, and it is called as lagging strand. They are called as lagging strand, or they are called as discontinuous strand. Discontinuous strand, and they are also called as Okazaki fragments. Okazaki fragments. So these Okazaki fragments are united by the DNA ligase. That is the main function of the DNA ligase. Let us suppose these are the Okazaki fragments. They are Okazaki fragments. So these Okazaki fragments are united by the DNA ligase. After uniting Okazaki fragments, a continuous strand will form now here. So that is the main function of DNA ligase. Remember, DNA ligase is not involved in leading strand. It's only involved in lagging strand. Why it is involved in lagging strand? Because the strands that are formed not continuous, but they are discontinuous strands are formed. So it is compulsory to unite them to form a continuous strand. That function is done by the Okazaki DNA ligase. So Okazaki fragments are united by DNA ligase. So DNA ligase does not involve with the leading strand, it involves only with the lagging strand. And similarly, here also in case of the recombinant DNA technology, as the two complementary strands, when they get separated, when they get united, the sticky ends, I mean these ester bonds are formed here and here. They readily unite with each other by the DNA ligase. So remember always, a DNA ligase will unite one fragment with other fragment of same DNA strand. It does not unite one strand with other strand. It unites one fragment of one strand with other fragment of same strand. You are seeing here, all they belong to the same strand of the DNA. All they are united here. Right. Let us suppose this is first fragment, second, third, fourth. All these four fragments are united with each other by same DNA ligase. And DNA ligase is not joining, it's not uniting this strand with this strand. Let us suppose this is strand A, this is strand B. It's not uniting strand A with strand B. It is uniting DNA fragments of same strand A. So that is the main function of the DNA ligase. Now let us see here the question. Identify the wrong statement with regard to restriction enzymes. Each restriction enzyme functions by inspecting the length of DNA sequence. Yes, because when you provide the DNA to restriction endonuclease, first it will scan, it will inspect all the length of DNA to check whether any palindromes are there. They cut, second statement, they cut the strand of DNA at palindromic sites. The restriction sites are where this called restriction sites or cloning sites at which they are cut. They are useful in genetic engineering. Exactly, because without molecular scissors, you cannot imagine the recombinant DNA technology, because with the molecular scissors only we cut the vector DNA as well as desired DNA and we unite them with the DNA ligase and we make up a recombinant DNA. This hybrid DNA is introduced into a bacteria and that bacteria is allowed to attack upon a plant or animal where the desired DNA get expressed. So the third statement is also very much correct. Fourth statement, sticky ends can be joined by using DNA ligase. No, sticky ends are not joined by using DNA ligase. In fact, here the ester bonds are joined by the DNA ligase. So single strand broken fragments are joined by DNA ligase by catalyzing the formation of ester bonds in between the sugar and phosphates. So this statement is wrong here, because DNA ligase does not form, it does not join the sticky ends. So this is the question here. This question is very tricky. One has to understand it very clearly, because sometimes all the four options appear to be very clear, but upon careful observation only you will understand that fourth statement is not correct. So this is my explanation. I think it is helpful for you for better understanding of the concept. Please drop your comments, drop your responses in the Vivid Botany comment section. I will be responding to your all comments, and if any doubts are there for you regarding the technology, recombinant technology or somatic hybridization or any part of the botany, I am ready to explain you each and everything with detailed elaboration. So thank you very much. Please become a subscriber of my channel Vivid Botany. We will be discussing all important questions, tough questions of the NEET, previous NEET question papers. And if any doubts are there also, I am ready to discuss with you. Right? So thank you very much.